It's my pleasure to introduce our speaker today, Dr. Sean Hoban. He is a tree conservation biologist at the Morton Arboretum in Chicago, USA. Sean also has a leading role at the International Union for Conservation of Nature, Hoban, and the Coalition for Conservation Genetics. He has a PhD from the University of Notre Dame and performed a postdoc with the European Conservation Knowledge Dissemination Project and another postdoc at an NSF-funded mathematical biology synthesis center. He has more than 80 publications and currently focuses on developing science-based advice on ex situ collections like botanical gardens for understanding the genetics and ecology of threatened species to guide action and for bringing knowledge on adaptive capacity of populations to national and global policy. He has helped develop and deploy several methods for assessing genetic diversity in botanic gardens and seed banks, and he regularly works with organizations like the Center for Plant Conservation, Botanic Gardens Conservation International, U.S. Forest Service, and U.S. Fish and Wildlife Services. He also has interest in seed dispersal, macrogenetics, hybridization, and population size change over time. The title of his talk today is Making Ex Situ Plant Conservation More Effective, Conserving Genetic Diversity Using DNA, as well as simple, affordable proxy metrics. Welcome, Sean.
Thank you so much, Yanni. Thanks everyone for attending today. Hope you can see my slides okay and that you can hear me okay. If there's any problems, just let me know, please. That was a great introduction. It's a real pleasure. My name is Sean Hoban. I work at the Morton Arboretum, which is a botanic garden outside of Chicago, and I'll be talking about how we can both monitor and report and improve the genetic diversity that's in our ex situ collections. I want to first acknowledge that the Morton Arboretum is located on the ancestral homelands of the Ojibwe, Ottawa, and Potawatomi, and other tribes that resided on or migrated through this land for generations, including the Illinois, Miami, Sauk, Fox, Kickapoo, and Dakota, and I acknowledge and respect the traditional caretakers of this land and their ways of knowing. I'm here as a lover of plants. I've always been fascinated with their structure, their ability to dominate and form entire environments, their biochemical and physical adaptations to harsh environments. I'm here as a conservationist. I'm very concerned with the fact that about 30% of our trees are threatened with extinction. Due to causes that we know that we have documented over the years, logging, conversion of land to mining or urban areas, conversion of land to agriculture, and pests and diseases. And I'm here today as a member of the Global Botanic Garden community and ex situ community. Botanic gardens are an important partner in conservation. They have unique resources in terms of knowledge and expertise in horticulture, data management, science communication, plant exploration, and many botanic gardens have in their mission a commitment to science education and conservation and solving global challenges like food security and biodiversity loss. The Morton Arboretum is a fairly large botanic garden, both in terms of area and attendance. We have over 1 million people who visit every year and tens of thousands of students who take part in our programs. We have active on-the-ground work in hundreds of communities in the Chicago region, helping people take care of their trees, develop policy and training. We have a Center for Tree Science in numerous areas of plant science. We have a tree breeding program developing regionally adapted and resilient plants for release. And we have a science communication and global conservation action program. In my team, we developed a mission about 3 years ago, and our mission is to provide the knowledge and tools needed to guide conservation action, focusing on resilience and adaptation in a changing environment. We particularly focus on genetic conservation, both in situ and ex situ. We also particularly focus on developing metrics, measures that are easy to deploy to assess how well we are doing. How much progress have we made with conservation? How do we set new priorities? And we work with policymakers, and I have an emerging interest, not expertise by far, but an emerging interest in health, plant health, and disease. In my team, and I'll emphasize this throughout the talk, we don't just do research. Most of my work these days is really applied research and is connected to outputs that can be used by other people. So we summarize our work in policy briefs, in communications to the botanic garden community, communications to the public, and establishment of best practice guidelines. We are all here today. Well, many of us are here today because of the challenge that we face in terms of trying to conserve the genetic trait and ecological diversity of plants across their native distributions, as we are also faced with limited time, money, resources, and space. So I think we're all interested in developing new and better tools and adopting those tools that can be used to assess and report on our progress and make more efficient use of our resources. We've been working as a gene bank community for over 100 years, but the first, to my knowledge, quantitative efforts to provide advice, to provide a set of tools for saying how much we need to conserve was developed in the 1970s, but for example, by Brown and Marshall here, who noted that we have limited space. So how do we calculate what is the minimum amount of seed, of plants that we need to keep ex situ to have an efficient maximal amount of genetic diversity? They really set the course of research for the next few decades, and I have followed in their footsteps, and my work and the work of others has shown that that we do need to adapt our sampling, our protocols, our goals to each species biology. So each species dispersal, their size, their longevity, their geographic distribution, their commonness, all of these will influence how much and how we should preserve them ex situ. And I'll show you some examples of that today. So today I'm going to start with 5 to 7 minutes about the Global Biodiversity Framework. I was invited to talk and to place my work in the context of the Global Biodiversity Framework. So that's where I'll begin. I'll then look backwards at previous reporting under the Convention on Biological Diversity, because I think that holds important lessons for how we can report in the future. And then I'll spend about 20 minutes talking about particular techniques, about 5 minutes on each of these. What are some of the various tools we can use to assess and report on genetic diversity? The Kunming-Montreal Global Biodiversity Framework is the latest iteration of the Convention on Biological Diversity commitments. This is the treaty first created in 1992, signed by almost every country in the world, and in force since 1993. It commits those signatories to 3 endeavors: conservation, sustainable use, and fair and equitable sharing of benefits. And one of the parts of the convention is to report periodically on progress. So about every 4 years, countries are required to produce a report on the state of biodiversity and how they have contributed to the global targets. Prior to 2020, the CBD had committed to a decade on biodiversity from 2011 to 2020. Those were the so-called Aichi targets that, of course, expired in 2020, and countries wanted to commit to a new set of targets, recognizing that those were not completely successful. So the new Kunming-Montreal Global Biodiversity Framework will go up until 2030. It has a longer-term vision to 2050, and that vision is a world living in harmony with nature, a healthy planet delivering benefits essential for all people. It's a grand vision. Beneath that vision are several goals. Part of one of those goals is on genetic diversity, and then there are 23 targets. One of those targets has a component on genetic diversity, which is mostly what I'll focus on today. So although we'll focus mostly on that target 4, where genetic diversity is explicitly mentioned, it's worth noting that genetic diversity is an important tool or a concern for many other commitments in biodiversity. So these are some of the other targets in the Kunming-Montreal framework: invasive species, sustainable use, ecosystem services, restoration, spatial planning, and protected areas. Genetic diversity can be used to guide or is a particular concern for some of these issues. So let's focus on that target 4. I want to just unpack a little bit, explain a little bit of this wording. To maintain and restore genetic diversity. It says 2 parts. The first part, maintain, means to not lose, but to restore means to perform actions that can benefit or restore evolutionary processes, that can restore adaptation, ability, such as reconnecting populations or supplementing populations to a given former population size. Within and between populations, these are the 2 components of genetic diversity within populations, is those within population processes, inbreeding and the speed of local adaptation to like local changes. Between populations is that diversity distributed among populations, and these are all the different traits, the range of conditions a species can persist in throughout all the environments it occurs in. Both of those are important for species survival. Native, wild, and domesticated species, that's all native species. And maintain adaptive potential means that's the reason why we are conserving genetic diversity, to help species adapt to a changing environment. It took a long time to negotiate this agreement from late 2019 up till the end of 2022. In the so-called zero draft of the framework, there were actually no mentions of genetic diversity in the targets, and the goal was not very ambitious and not super coherent, actually. And it was a long road of negotiations, of many meetings and correspondence to make the target more specific and ambitious. And if you're interested in reading about that policy journey, you can check out this paper here. My colleagues and I were very active in this policy development. So we did a lot of scientific publications on this topic in terms of what is the state of knowledge on genetic diversity, what is the implication of different potential wordings and indicators. But we also participated in dialogues with people attending the CBD meetings through webinars. We produced and distributed several policy briefs on this topic. As you can see here, we created those in as many languages as possible so that they could be uptaken globally. Every time there was a new draft of the CBD, my team and I would submit comments, suggestions, and then lastly, we attended the COP15 meetings in Montreal. And this is a side event we held to present on the latest knowledge and tools for genetic diversity. And then we also had a booth at the convention so people could come and learn about and ask about genetic diversity. I think this was much more active engagement than geneticists have ever made towards the CBD or other national and international policy commitments. And I think it really did help reshape the wording of the target and also the understanding of policymakers on this issue. So that is the background, the Kunming-Montreal Biodiversity Framework, how it came to be. I told you a little bit about the wording that countries have committed to. So let's now look backwards. How did countries report in the past? So in that 2011 to 2020 period, we had the Aichi framework. This was the Aichi target 13 on genetic diversity. You can see it was mostly focused on cultivated species and their wild relatives and other socioeconomically and culturally valuable species. It says that their genetic diversity should be maintained. Also, strategies should be developed and implemented for minimizing genetic erosion and safeguarding genetic diversity. So that's what countries committed to in 2011. How did countries actually report on that? Well, my team, we analyzed 57 countries' national reports over 2 reporting periods from 2014 and 2018, the most recent cycles. The next one will be due in 2026. We tried to get a pretty global representation of countries in this analysis, both in terms of geographic distribution, languages, and income levels. We read each of these reports thoroughly and did kind of a textual analysis where we looked for different mentions of genetic diversity with respect to its status, threats, actions, obstacles, and progress. A big caveat is national reports are summaries. Many actions, threats, etc., present in the country don't make it into a report. A report is limited by the report writer's knowledge, time, capacity, and access to data. But by analyzing these reports, I think we did get some insight into what were the major concerns and what were countries paying attention to with respect to genetic diversity. Countries did note the importance of genetic diversity for these main issues: resilience to environmental change, productivity, and developing new varieties. Most countries had a national level target somewhat equivalent to this Aichi target, but 20 to 30% of countries actually did not have genetic diversity mentioned in a target, even though countries set national level targets analogous to the global target, and they're supposed to be pretty well aligned. So it was concerning that many countries were not including such a target. And a very small proportion of countries were including this part about strategies for minimizing genetic erosion. So we were looking to see if countries had developed some kind of national strategy or plan, which was called for in the target, but most countries were not doing that. So what kind of actions are countries taking, or were countries taking during the previous decade to support genetic diversity? Most common was investment in seed and gene banks, then investment in breeding or research agencies, and then advancement of law or policy on genetic diversity. And this was consistent. This is 2014, this is 2018. Much less, at least mention and discussion of actually assessing that genetic diversity, either at a time point or over time, less investment in training and education, or at least less mention or description of those activities, and less in terms of in situ funding for genetic conservation projects. We then looked through these reports for any time a particular species was mentioned. When we found a species mentioned with respect to genetic diversity, we put it into one of these categories based on what the report, the context of the report, and whether it was about a threat, a status, change, or an action. You can see that crops and farmed animals were by far the most frequently mentioned in these reports with respect to genetic diversity. Interestingly, wild relatives of animals were very infrequently discussed, but we have some discussion of crop wild relatives, forestry, and species of conservation concern. I think it's worth noting that these reports were not giving much space to or attention to wild harvested species, species providing ecosystem services, and culturally important species, even though the Aichi target did ask for countries to conserve genetic diversity of, for example, culturally valuable species. So definitely a strong bias in the types of species countries were reporting on with respect to genetic diversity. So overall, in analyzing these national reports to the CBD in 2014 and 2018, we conclude that in the CBD, the largest and most comprehensive instrument for directing biodiversity conservation globally, parties focused on a small set of genetic conservation actions by a subset of species and insufficient indicators. Going forward, we had a few recommendations in this paper. You can find more by visiting the paper. Each of these QR codes links to the particular paper that I'm discussing. Countries should, in the future, report on genetic diversity with respect to all species. Of course, it's valuable to highlight particular subsets, whether it's pollinators, whether it's crop wild relatives, whether it's species of concern. Of course, very useful and important to break out and understand the genetic diversity of those groups, but we should not overlook the genetic diversity of all the rest of species. There needs to be greater attention and description of the state of genetic knowledge in countries. We need this to understand their capacity gaps, greater attention paid to linking in situ and ex situ activities, and also an understanding of local knowledge and use of genetic diversity. There continues to be confusion among policymakers, and understandably so. Genetics can be a complex topic, but we do need improved definitions and glossaries for terms relating to genetic diversity. What does genetic erosion mean? What does safeguarding mean? What is within population genetic diversity? And to do that, we need greater communication and collaboration across the science policy, sometimes divide. And then again, I would highlight, I think this was a neglected part of the Aichi target, was developing national level strategies or frameworks for conserving genetic diversity. So what I did there was I introduced the Kunming-Montreal Biodiversity Framework. I talked a little bit about the Aichi framework, which preceded that, and how countries reported in the past decade. So now let's look forward and talk about how gene banks and ultimately countries can assess and quantify and report on genetic diversity they have conserved. I'll mention 4 approaches briefly, about 5 minutes on each. That's DNA-based studies, simulations, geographic gap analysis, and a little bit more speculative here, recent concept I've been thinking about called effective size. So the most direct way to assess genetic diversity in a gene bank or in a garden is to actually collect samples from the individuals in that collection, go out into the wild and collect samples from the wild populations, and then analyze this in a DNA lab. So you're going to quantify all the genetic variants that exist in the wild, quantify what subset of those are present in the ex situ collection. So that's just a fraction of the wild that is preserved ex situ. I'll first talk about rare species. I will get to some examples later of common species, but most of my work has been a little more on the rare or threatened species side. These are all threatened species in botanic gardens in some very high profile genera, such as hibiscus, magnolia, quercus, cycads, and palms. This is the number of plants currently in what's called the meta collection across all botanic gardens. And this is how much genetic diversity is conserved in those number of individuals. And 3 out of the 11 of those meet, or almost meet, a benchmark set by Marshall and Brown in the 1970s and still considered a fairly good goal for an ex situ collection, which is 95% of the genetic variants present in the wild being conserved ex situ. So the remainder of these species have not met that goal. If you plot those points, it's pretty clear that as you collect more plants and get them into ex situ collections, you're getting more genetic diversity. It is a nonlinear relationship, so this kind of levels off. Between 100 and 200, it starts to level off. And so perhaps going forward, a good benchmark for rare species would be collections in the range of a few hundred plants. So this is one thing we can do with DNA analysis. I want to emphasize that DNA can also tell us other things about a collection, which are not discussed as frequently. So it's mostly been about, like, do we have enough of the genetic variant ex situ? But you can also understand other aspects of your collection. And I'll talk about the case of the maple-leafed oak. This is only found in 4 populations in Arkansas in the US. We analyzed the ex situ collection relative to the wild, and we found that the ex situ collection has a very high proportion of full siblings. This is not surprising. We usually collect seed from a few maternal plants, and so a lot of the seed should be siblings. But this could be a problem for future collections if you then want to use that as a breeding stock. If most of your individuals ex situ are related, you could have inbreeding problems in the future. We also assess redundancy. Of course, it's important to back up your collection. So ideally, every genetic variant should be present in 2, 3, 4, 5 copies, depending on the degree of risk or probability of disaster. This particular collection of maple leaf oak, not more than 95% were already present in at least 2 copies. That's very good. As you get to a really high number of copies, that would be a really catastrophic disaster, would have to remove that many individuals. But again, we can quantify how many genetic variants are actually backed up multiple times. And then lastly, representation. So you can imagine a species occurs in distinct populations. Some of those populations might have been collected from better than others. Sometimes we have data in the passport or accession data about where that seed came from, sometimes not. And DNA can actually be used to match sometimes an individual back to its source population, even if we don't have good accession data about where it came from. So for this species, we matched all the XC2 individuals back to their wild population, and 3 of the 4 populations had really high sampling. One population had only 6 individuals at Situ, all of them at one garden, Chicago Botanic Garden. So this DNA analysis highlighted that we really need to sample more from this particular population. So these are some other things that DNA studies can tell you that also complement that percent of genetic diversity conserved. So the main message is DNA studies can quantify how much genetic diversity is conserved, but also identify other aspects relevant to managing and enhancing a collection in the future. So now I'll move on to simulations. Genetic studies are still fairly slow, they're still fairly expensive, and they still require technical expertise both to conduct and to interpret those results. Can we use something that is faster and cheaper? Simulations are one way to do that. So my team, over the past 10 years, have developed this approach to use simulations in the absence of any genetic data, which makes it essentially free in terms of just needing person time and computer time, and not expeditions, DNA supplies, sequencing, etc. It has its caveats. Each of these approaches has major caveats. So what we do is we take the species geographic range, we simplify it down to a picture model like this, with each population and its approximate size. We simulate this model, and then we simulate sampling from that model, and then we quantify how well each sampling did by comparing our simulated seed to our simulated wild model. So it's similar to that DNA-based study, but all taking place on a computer. So this might be our model. We're going to just take samples from 1, 2, 3, 4, etc., up to some large number of samples. And each time we take a sample, we can compare our sample back to the wild and get a percent of genetic diversity conserved, and then use this threshold of 95% and say this is our minimum size. So this is what it looks like in terms of a graph. We're taking a number of individuals. Every time we get that simulated collection, we get a percent conserved. At 95%, we say this is the minimum sample size. Here it would be about 80. So we did this for 14 threatened oak species in the US. In the time it would take one genetic study to perform, we simulated in pretty good detail 14 threatened oak species geographic ranges on a computer, sampled from them. This is showing the 95% threshold and where each of these lines crosses. And for most species, most of these oak species, the threshold minimum sample size was between 200 and 450. So this is a little bit higher than what was recommended in our DNA-based study. I can explain why if anyone wants to know. But it's still fairly consistent in hundreds, but not thousands of samples are needed to conserve these rare plant species ex situ. Okay, now I'm finally going to talk about a common species. This can also be done with a common species. Simulation can even simulate millions of individual plants across this geographic range. So European ash has been disappearing for several decades due to ash dieback. Kew Botanic Gardens and partners in the UK wanted to conserve the genetic variation of this species across the UK before it was lost to this disease, which kills 95+% of trees. So they collected several million seed, and we partnered to try to evaluate how good that collection was and how it could be improved. So this is actually our simulation model. It looks very much like the outline of the UK because it was parameterized based on actual forest inventory data. So you can see, darker spots are where the species is more common, more abundant. So we simulated this model across the UK, 1,000 individual populations, and sampled from it and came up with a couple on the ground useful recommendations. The first was, what's a good stopping point? Where, as you go along this curve, which I've shown several times, all of these curves you've seen start to flatten out. At some point, the slope of this line is very close to zero, very close to flat. And you might say some arbitrary threshold, I should stop sampling when it's close to zero. In this case, 0.5%. This is just reflecting as you invest more money, you keep getting less and less return. As you invest more time, more resources, you get less and less return. So we defined a stopping point, and that was about 30 populations and 30 trees per population. Interestingly, the foresters a priori had said, we think it should be about 50 populations and 10 trees per population, which is in the ballpark of what we found based on our actual simulation. So it was a really great partnership to combine this kind of a priori forester knowledge with the simulation. I'm often asked, is it better to sample more seeds from a given plant or more plants on the landscape? All else being equal, it's always better to sample more plants on the landscape. After you get to 20, 30, 50 seeds from a given plant, you're not getting any new additional genes from that seed set. So that flattens out pretty quickly. Whereas sampling from more plants, you're going to get more genetic diversity. So this is showing 50 seeds from 10 plants or 50 trees with 10 seeds per plant, much more genetic diversity. So across all our research, it's much more impactful to go farther out onto the landscape and get more sample from more plants. We can also, with this simulation, test geographic sampling. So we had these very kind of iconic sampling strategies around the edge or the core, or sampling on a grid or from each of the seed zones in this case. Which of these performs best? You can probably imagine these, which are distributed across the geographic range from north to south, east to west, perform best. Just behind them, and not too far behind, is focusing the sampling on the core of the range. There's a lot of caveats to this conclusion, but it is important to show that simulations can be used to at least compare these different strategies. And we can even do that at a local level. So this is simulating within a population. Of course, all the manuals, all the intuitive advice would say you should sample in this fashion, which covers more of the population than, for example, here, where you've just parked your car or had your trail lunch and sampled the 10 trees closest to that. Of course, these should perform better, but simulations allow us to actually quantify how much better. So the blue and yellow are these top two. Pink one is this biased sampling by the side of the road, and this is your transect sampling in green. So more than 50% more genetic diversity would be collected in this case from sampling more widely across the population. Maybe you have limits. Maybe there's a private landowner here and you can only sample this much. Simulations can tell you how much approximately genetic diversity you'd get if you had that constraint. So I hope I've shown you that simulations give us a lot of neat ways of testing geographic sampling and improving our ex situ collections, both in terms of providing advice on minimum sampling and sampling logistics. So now I'll mention a different approach that is even more scalable and even faster, but has different caveats. So we can use GIS-based tools. This is the so-called gap analysis method, which some of you may be familiar with. This is just, we take a map of wild locations, we place on top of that a map of where we have sampled seed, and we get some measure of coverage, how much of that wild distribution has been sampled from. This was originally proposed at least as early as 20 years ago by Sally Aitken and colleague. It's really been pioneered by Dr. Callan Curry and his colleagues, and we are really following in their footsteps. The white dots show the species geographic range, and the black circles show where we've sampled from. So the area in black divided by the area in white, this is Georgia oak in the United States, is the geographic coverage. And then the ecological coverage is just the number of ecoregions, the colors species occurs in, and which we've actually sampled from. What is so useful about this approach is it's highly scalable. Once you have these points, assuming those points are cleaned, reliable, accurate, have the right species designation, you can scale this up to essentially as many species as you want. We've done this for a few dozen oak species. This puts species into buckets of these are well conserved and these need a lot more sampling to fully conserve them. And so this helps us set priorities, like these are higher priority than these. It also helps us to track progress over time. These are more recent collections, and you can see we are helping it fill in some of that unsampled geographic range. Acknowledgement to the U.S. Forest Service and American Public Gardens Association that fund a great program called the Tree Conservation Program. And I've done this myself. We did this for Quercus havartii, a desert oak. This is actually an oak tree in Utah. And previously it was only present in one botanic garden from one collection. And so we really expanded the geographic coverage in ex situ collections of this species. It was one of our priorities from over here. So we've done this for a number of other tree genera in the United States, pines, walnuts, laurels, yews, hickories, beech, and a few others. Again, I have to acknowledge and really thank and just all of the appreciation to Dr. Colin Cowrie and Dan Carver and their many dozens of colleagues who really pioneered this approach almost 10 years ago and have deployed it at an even greater scale than we have at the Morton, including hundreds of crop wild relatives in the US and thousands of human-used species globally. In the US, they pointed out that we have a really urgent need for action for getting our crop wild relatives conserved, and globally, it's even worse. This is the conservation status ex situ. Almost no countries are out of the red, and this is in situ protection. So how much of the range is covered by protected areas? I think the Natura 2000 network in Europe is contributing a lot to this green area. So both in situ and ex situ collections are not protecting sufficiently species geographic ranges, with a special need focused on expanding the geographic range conserved ex situ. And this is a publication by Dr. Cowrie and his colleagues. So gap analyses are highly scalable. They can identify which species have much of their geographic range coverage and which have not. And they can also track progress and point to quantify progress over time. So the last tool I want to mention is a little more speculative. It's this concept called effective population size. Is there something else we can use that can quickly, affordably, and in a standard fashion assess how our ex situ collections are doing? The Kunming-Montreal Agreement has about 6 indicators on genetic diversity. 2 of them are shown here. These are mostly designed for wild populations. One of them is the proportion of populations with an effective size above 500. This is considered a sufficiently large population in which Species can adapt, they don't become inbred, they maintain their genetic diversity with essentially no loss, essentially no genetic erosion over fairly long periods of time, of at least dozens of generations. So this concept has actually been used in forestry, in fisheries, in agriculture, in conservation biology for several decades, and is a fairly well understood concept among actually a lot of practitioners. How do we calculate effective size? We can get it from genetic data, or in a wild population, NE is about 1/10 of the census size, adult number of individuals. For gene banks, I think NE would approximately be the number of maternal plants sampled, because the maternal line is the limiting factor in effective population size. So comparing the number of maternal plants sampled to this effective size might be a useful indicator for gene banks. We've been deploying this indicator for wild populations in partnership with 9 countries' biodiversity or wildlife agencies, and it's been actually pretty successful. We have data for wild populations for over 900 species and thousands of individual populations on effective size gathered from a large diversity of sources. We've developed a pretty good methodology, standard way of storing this kind of data. We can even include qualitative information. So we have deployed this indicator for wild populations, and it's being uptaken as an indicator for the global biodiversity framework. Could we do it for ex situ collections? I think so. I think we could, again, use the number of maternal lines as a standard way to assess the genetic condition of hundreds of populations and species in gene banks using passport data, accession data. And this could be another tool, again, for prioritizing species, quantifying progress over time, and connecting, I think importantly, connecting genetic diversity to the public and policymakers. These simple metrics can be actually visualized and understood by non-geneticists, non-scientists, and can actually be quite meaningful measures. So have not tested this, have no data, but I think this could be a neat indicator to explore for gene banks. Botanic Gardens Conservation International has a tool called Plant Search, and in a lot of cases, we do have the number of maternal plants, and I'm interested in exploring this in the future. I want to leave you with a dilemma, though, before I close. We don't know how much diversity plant species need. The amount and type of adaptation genetic diversity needed is totally unknown. It depends on what happens in the future, how hard the environment changes, and what new things arise. Plant disease resistance may occur in 1 in 10, 1 in 100, or 1 in a million plants, which of course says we would maybe need 90%, 95%, 99%, 99.999% of the diversity sampled. This is a really hard problem that needs future work. So I want to thank the people who've supported this work. In particular, Institute of Museum and Library Services have really been supportive of our work in improving botanic garden collections. U.S. Botanic Garden, American Public Gardens Association, NSF has supported several of our works, including a training grant for young researchers, and of course, the Morton Arboretum. So I've shown you genetic diversity as a key part of the Kunming-Montreal Global Biodiversity Framework. I showed you how we looked back to learn lessons about country reporting on genetic diversity in the previous iteration of the CBD. And I talked about these 4 different tools: DNA-based studies, simulations, geographic gap analysis, and effective size, as ways to quantify our collections. And with this diversity of tools, we can quantify progress, produce on-the-ground advice, guide the prioritization of species and populations, and all while using diverse data. And as a team, we can work towards better documented, more diverse, representative, and backed up collections of plant biodiversity. If you're an early career person, there's lots of opportunities in my group and other people's groups. I can help connect you to those in terms of internships, in terms of undergraduate opportunities, or recent undergraduates you can check out our lab website. And I do mentor students at the University of Chicago for people interested in doing graduate work on this problem. So with that, I would just like to thank you, and I look forward to questions.
Thank you very much, Sean. That was great. And we do have one request and one question, and I'd like to read that question. Can you please share any examples of cases when genetic diversity in ex situ collections were strategically sampled to reintroduce plants to the wild? So do you know off the top of your head if there was any case in which that happened, genetic diversity was reintroduced into the wild?
The one example that first comes to mind, the first example is with that maple-leafed oak, which is a rare tree species that is being conserved ex situ, but those ex situ collections are being built with the express purpose of producing seed in those orchards and to use for reintroduction. The same is being planned for white bark pine. White bark pine is a very widely distributed keystone ecosystem forming species in the US Western, US and Canadian West. That species just has been really impacted by a disease, but seeds are being collected of that species, being guided by genetic knowledge through the US Endangered Species Act recovery planning process, and those seeds are already, at least sites for reintroduction are already being identified.
Wonderful. I have a question about number of sampled plants or individuals that you are collecting. And you're very fortunate because you have trees and you actually can go back to the original population because it's a good chance that that tree is still alive, or at least, you'll find a population that's very similar to it. When working with crops, we have some standards that say that for cross-pollinated species, we should have, the minimum is 30 individuals should be collected to contain 95% of the alleles that you need for that particular accession. And for self-pollinated, you're looking at collecting 60. So it's interesting to note that some of those numbers actually fall within those curves that you showed of number of individuals that you need for the above 80% of the genetic diversity that you should collect in order to get, the full amount of alleles that you wish to have, so that if there's anything that you need, that you would find them in there for health reasons, plant health reasons. So have you thought of doing some of these studies with cultivated crops? But I'm wondering what the challenge would be there to find the original populations in order to be able to compare how much diversity you actually have in your gene banks compared to what people collected, say, 50 years ago. And see if those numbers from 30 to 60 change. If we need to up, for the most part, you say you have to bring home more, you have to collect more, you have to have more, upwards of 120 and even 500, right, for some collected things. So how would we go about modifying these simulated? By the way, these two numbers come from simulations. So how would we go about finding a more exact number that then can be put into the standards and best practices so that we can say for cultivated crops, these are the two numbers we're looking at to best capture all of that diversity?
Sure. The numbers, between 30 and 60, that number goes back to Doctors Brown and Marshall from the 1970s and 1990s when they worked on that problem. And I still think those are good benchmarks for each local population. So that's the number of plants to get each local population's diversity. That's still pretty reliable benchmark. What I showed you in terms of simulations is if you know a little bit more about the population sizes, the geography of this species, or its mating pattern, you can get a little more exact. I should note that those numbers also, are looking at more common variants and are excluding like really rare variants, which may be important for disease. Simulations can be used for, again, tailoring that to specific species biology. They could be used to try and answer the question of what do we need for a like really rare disease resistant variant. Um, I still think those numbers are good benchmarks to start with if you have nothing else available and you don't have a DNA-based study or time to do a, a computational study. Those are still reliable. Again, if your goal is not to get the most rare variants, this comes back to this challenge of we don't know what kind of genetic diversity is needed for the future, which is a problem that I think can be worked on and should be worked on soon.
Okay, wonderful. We have the next question is, thank you very much, sir, for a very informative PowerPoint. I need your suggestion and comments regarding on in situ conservation. What are the best practices and approaches that you would make regarding in situ conservation? You said that that was something that was not as well covered. What are some of the things that you think should be done in situ then?
Sure. Uh, the two most important things to be done in situ relate to those indicators that I mentioned under the Kunming Montreal biodiversity framework. The first being make sure individual populations do not go extinct. Each individual population may contain unique adaptation to that local space, um, or rare variants due to some disease. So that's first, is make sure each individual population does not get lost. And the second is try to increase population size above that effective size of 500 or as close to it as possible. That can take many decades for some species, but that's a goal to shoot for. That population size metric will mean the species has sufficient variation, sufficient chances to undergo natural selection and actually adapt to even fairly rapid changes. So those are the two things. Don't lose populations and get those populations through supplementation, through management of the habitat, through replanting, whatever is possible, boost those numbers. The third thing is if it's a really high priority species, and as DNA technology gets cheaper, you can, monitor those populations over time and identify things like I mentioned early on, inbreeding, see if that is a problem for the population. Those are the things.
Okay, thank you. Next question. Is it feasible and how much does it make sense to compare the genetic diversity estimates of different species generated by different DNA marker systems, for example, SSRs or SNPs?
A good and detailed question. I forgot. To the previous question, another important thing in situ is for those populations that have been drastically fragmented and they were previously connected, reconnecting those to allow gene flow and to allow those variants that are adapted to different parts of the range to kind of move, especially as climate changes. That is also very important. So reestablishing connectivity. As for use of different DNA markers, there are differences in the actual raw values, depending on the study, if it uses a different marker. Um, as we move towards whole genome sequencing and just large genome sequencing in general, most studies will be pretty comparable. Our own work, including a paper that just came out, did show, yeah, there are differences between SSRs and genome sequencing. But if you have several studies and as more studies have genome sequencing, basically most of our work will be comparable among species. So definitely a concern, but in the future, most species are going to be using a pretty comparable type of sequencing.
Yeah. And as a follow-up to that, to enhance the global collaboration and support to assess the genetic diversity of forest trees, is it feasible to come up with a common protocol of DNA and marker analysis adoptable for developing countries as well? So is there a recommendation in the incoming that would kind of standardize? You talk about standardizing ways and ways of reporting. So would that be one way of doing it? A common protocol for DNA and marker analysis?
I think that is a great goal to shoot for. The DNA sequencing processes are becoming standardized, but it's the analysis part that has been very hard to standardize. I think that's an important goal to shoot for, and I know there are several groups working to create more easy to use, replicable analysis pipelines. That's a big challenge. I think it is a really important goal, and that would alleviate a lot of the required technical expertise. So I think that's an important goal to be aimed for. I would say, so I'm trained as a geneticist. I think DNA data provides really valuable advice. But when we're looking at conserving tens to hundreds of thousands of plant species, these more simple, faster, proxy-based tools are also really important and should be considered alongside DNA.
Yeah. And related, third part of this, given the crucial role of genetic diversity in species resilience, could you explain how emerging DNA technologies might be integrated with cost-effective proxy metrics to monitor and manage genetic variation within ex situ plant conservation efforts, particularly in rare or endangered species?
Um, I think that was that's the main message of my talk, I think, is that both approaches are useful. Sometimes you will have the funds, the time, the expertise to do detailed DNA studies of particular species. But I think even for the next decade, 90 to 99% of species, we don't have the logistics even to get to the plants in the wild to sample them thoroughly and understand their DNA in the wild. Um, so the cost in the laboratory is one thing, but another is the expedition that has to go out and find the wild plants and characterize those pretty well. And that increasingly is actually a lot of the cost. Um, so I think to integrate these strategies is just to decide which species are you able to, both logistically or their high priority species, and do DNA-based studies. And for the remainder of species, do the proxy-based mapping, effective size, gap analysis, etc. And then for some species to do both and compare them. And our group is doing that right now, taking some species where we have genetic data and where we have these sort of simpler measures and comparing them.
Yeah. Yeah. Another question, should we be keeping seeds from different mother plants separately as a routine? You mentioned that part of the mother plants, etc. And I was wondering, or she was wondering whether you should keep the mother plants and the seeds from the mother plants separately as a routine, rather than lumping them together, which is usually what we do.
Ideally, and part of that depends on the purpose and how that seed will be used, and also the biology of the species. For species where the if the first of all, if the seed bank has the capacity to do that, of course, that's going to take a lot more manual labor and space to keep them separate. But in the ideal case, yes, because that allows you to, if needed, plan, the regeneration and mating of those seeds in the future. Um, if you found also, it can help you track back to an individual plant if you find a disease variant, resistant variant, or a plant with a particular trait. If you have that seed categorized by maternal line, you can feasibly go back to that maternal plant if it's a multi-year, not if it's an annual plant, but if it's a longer-lived species, you can do that. Um, on the other hand, there are certainly going to be cases where the plant tolerates, inbreeding really well, or we just don't have the space and logistics to keep them separate. So that can be decided on a case-by-case basis, but there are advantages to keeping maternal lines separate.
Wonderful, thanks. What are the data requirements for the modeling approach?
Good question. Thanks. Um, the models we built so far are based on just geography and demography. So how many populations there are there, how separate, distant are they, and about within, a margin of error about how large are populations. Um, and we've even done, in one of the cases I mentioned, we did some sensitivity analysis, which is where you change the model a little bit and then see if the results change. And those models are fairly robust to like, oh, the population was actually double what we thought it was. That doesn't
Mm.
actually change the results that much. So you really only have to have an approximate understanding of the number of plants, number of populations, how far apart they are, to make those models pretty reliable. Um, we have not yet experimented with models based on, adaptive resistance to like disease or particular traits. And that's a real important next step, is to start building those models based on real knowledge of disease resistance genes.
Wonderful. And our last question, Sean, and this is a really good one. I mean, we don't know the answer to this. In your opinion, is there a reliable tool through which we can set a time frame for recollecting of the species in the wild to make sure we capture any change in gene diversity over time?
I only know of a couple of people who looked at this. Only one study comes to mind. There's a few, but Dr. Chris Richards at the USDA uh, and his colleagues have done this in a couple of cases and found that even over a few decades, there were not significant changes in a very large population of an annual species. Um, as climate changes more rapidly and as environments change, that interval might shrink. Um, or if a particular weather event happens. So I think it's safe to say a couple of decades would be fine in most cases, assuming the change is gradual. Um, but it has been shown if there's like a big drought event or a disease wipes through a population, really immediately after that event, the genetic composition will be very different. So I think a couple of decades should be fine, except in those cases where we know a big event has happened. And in such cases, the new population will have a very different genetic composition. And so a new collection is a good idea.
Yeah. Yeah, living in Southeast Texas, we were hit by three events. A hurricane, which had us underwater for more than 3 weeks, so plants had to survive to that. We had the worst drought we've ever seen in the last 20 years, and we had a cold spell we had never seen in 2021. So I can see from my window that there's a huge change because of those three things happening within the last 5 years. So 20 years is your recommendation, but if there are things as I have mentioned, drought, floods, or heat, that would have to be done a lot more, right? Maybe every 5 years, perhaps. Would that be your
Um,
recommendation?
or anytime one of those events happen. And
Right.
I would think that this is just speculating. I haven't thought about this before, but
Yeah.
off the top of my head, I think anytime one of those events happens, um, and I would wonder and I would suggest, if possible, that kind of information could be recorded in the passport or accession data. This was after a large event. So I think starting to record that information in our accession data would be really valuable to botanic gardens, to plant breeders, to focus on those populations where the individuals survived a certain event, and would have a really interesting component of genetic diversity. That would be, I think, really exciting to do. I'm not sure if that's currently done in passport data, but I think that would be really valuable.
Yeah, it would be. Well, with that, I think we bring the Grow webinar to a close. First of all, Sean, I'd like to thank you very much for putting on a presentation that is both very informative and provocative. Thank you very much. To the participants, thank you for attending and putting your questions up. We love trying to answer what you have on there and hoping that the information is useful for you. Sean, with that, I'll give the last closing words to you, and with that, we will close the Grow webinar.
Well, thanks everyone. I hope my work has motivated you to think about quantifying our progress, producing practical advice, prioritizing among species and populations, and using diverse data using both DNA and non-DNA data. And I look forward to meeting many of you in the future and working towards better collections of plant biodiversity.
Thank you very much once again. Thank you, everybody. Have a good time, and until next time.